I can't remember, but it's an easy way to remember that. I gotta find my notes on the intro to bio.
Yeah. Okay. Like a mnemonic device or something that you have.
Yeah, I remember because I had you for like lecturing the other lady for lab, but she told me to go away.
Well, if you find it, let me know.
I'm gonna go get it out my box.
Right. Okay, so substrate level, using the energy from breaking a bond. Oxidative phosphorylation is going to use proton motive force. This goes back to active transport as well. Remember, bacteria are the only organisms that make proton motive force on their cell membrane. And proton motive force was a separation of charges across the membrane, where the outside was positive, right, and the inside is negative. That's like a battery, right, having positive separated from negative. In this chapter, we'll talk about how it got that way, how the proton motive force was generated. But for now, we're going to assume it's there. And then to get energy out of proton motive force, you bring the proton back in, right, one at a time, and that brings energy with it. And if we use that energy to make ATP, it's oxidative phosphorylation, using the energy from proton motive force from the separation of charges across the membrane. The third way, this is the only time we're going to mention it, photophosphorylation, because it's only photosynthetic organisms like plants that do it. It's essentially the same as oxidative phosphorylation. We're still going to use proton motive force. The difference is light energy was what was used to create this proton motive force. The organisms that do photosynthesis use light energy to separate those charges, whereas oxidative phosphorylation, light's not involved. And we're not going to talk about photosynthesis in this chapter, so that's the only time you'll see that one. Oxidative and substrate level are going to be the two that we focus on. Questions on any of those? All right, so that was our first component. You gotta have ATP to get metabolism to happen. Second ingredient, an energy source, which basically means food, right? We need some sort of food molecule. As humans, we focus on organic things, right? Sugars, starches, fats. Bacteria can eat all of those molecules too and use them for energy. But they've got some options that we don't have. Most bacteria can also eat lots of inorganic molecules. We were just in my lab, you watched that little video. They talked about a lot of the sulfur bacteria, right, that can eat sulfur. There's some that can eat ammonia. They can use all kinds of things that don't have carbon in it for food that we certainly can. And they're doing very similar things. They're going to be breaking the bonds in those molecules, releasing energy. We don't have enzymes for using ammonia for food, but they do. Or at least some of them do, not all bacteria. Regardless of what they're eating for food, they're going to be doing redox chemistry, oxidation reduction reactions, which basically just means that we're going to be transferring electrons. A redox or oxidation reduction reaction means that electrons are going to be transferred from one molecule to another. Transferred from one molecule to another. In general, that means we're going to be taking electrons from this food and moving them somewhere else in the cell. So, you're moving electrons from one molecule to another. That means that one molecule is going to lose an electron, and another molecule is going to take it. So you've got two terms for that, reduced and oxidized. Reduced is the molecule that gains the electron. It picks one up. Oxidized is the molecule that loses the electron. Which I know seems backwards, right? Reduced sounds like it should gain the electron and it has lost it. Okay, I'm not sure why we chose reduced is the term, but I can at least tell you where oxidized came from. Oxygen is really good at stealing electrons from stuff. Okay, so oxidized comes from oxygen. If you had a metal fence, I've got chain link fence in my backyard that nobody painted for who knows how long, right? So what happened to my metal chain link fence? Rust, right? Oxygen from the air has stripped electrons out of the metal and left that brown iron oxide behind. Okay, so oxygen steals electrons.
You think it's called reduced because the electrons carry a negative charge?
Probably. Probably. Negative seems like minus. That's a good call. Yeah, that makes perfect sense. Okay. Happens to silver too. Right? If you have silver jewelry or real silverware, oxygen strips electrons from the silver, leaves tarnish, silver oxide, and it happens to chocolate, right? Old chocolate.
Is that the metal fence?
Yeah, Same thing.
And what is it, the answer?
Iron oxide for that one.
But it's oxidized?
It's oxidized. Yeah.
God, why I didn't know that last time.
Happens to chocolate. Old chocolate, you get that white powder on it. That's oxygen stealing electrons from the chocolate and leaving that little powder behind, which won't hurt you, by the way. It just
Okay.
doesn't taste very good.
I tried to
Yeah.
eat
That's
it, like,
oxidation.
maybe I can eat it.
No, you can still eat it, as long as it's not fuzzy, right? If it's fuzzy would be mold, but if it's just white powder, it's oxide.
So it's old.
It's just old.
Ew.
Yeah.
Someone's given me old chocolate before. That's gross.
Okay, so oxidized has lost the electron, reduced has gained. So the food molecule is going to be oxidized. We're going to reduce something else in the cell. Which brings us to component 3, the electron donor, the molecule that we're stealing the electron from, which again is food. So food is the energy source and it's the electron donor, whatever the bacteria has eaten. We're going to use it to break bonds and release energy as the energy source, and we're also going to steal electrons from it as the electron donor. So those electrons have to go someplace. That's our next component, number 4 there. The electrons that we steal from food are going to electron carriers. These are the molecules that pick up those electrons, and where they're carrying them to is going to be the place that makes this separation of charges, right? We need electrons to be negative on one side of this molecule. And so these electron carriers are taking the electron from the food over here to the cell membrane to generate proton motive force. So the electron carriers, specific ones that we use. Next slide. There are 3. NAD, these are plus signs, not T's. Right? NAD plus, FAD plus, and NADP plus. I wrote out their long chemical names, you don't have to know them.
Yeah,
Nicotinamide adenine dinucleotide phosphate, you don't have to spell that, but you do need to know the letters. If they've got the plus sign, that means that they're in their oxidized form. They're not currently holding on to electrons. They haven't gone to the food yet to pick them up. Okay? Once they have come to the food and they've picked up the electron, we write them with H's on the end. NADH, FADH, NADPH. Okay, and here's why. So let's say we had an NAD plus floating around in the cytoplasm of our bacteria, and that bacteria ate some glucose, some sugar. So the NAD plus is going to come over to this sugar and steal an electron from it. Well, electrons don't happen by themselves, right? Remember chapter 2, all atoms have equal numbers of protons and electrons. So when it picks up this electron, it's also going to pick up a proton. And in chemistry, the atom that has 1 electron, 1 proton, and 0 neutrons? Hydrogen. Exactly. When this NAD plus comes over to the food, essentially what it's picking up is a hydrogen atom. 1 proton, 1 electron, 0 neutrons. So that's why we call it NADH now, because it's holding on to a hydrogen, 1 proton and 1 electron. And that's true for all 3 of them.
So,
So we're going to take one of these 12 hydrogens away, essentially. We're going to break the bond attaching one of them.
So now they're in the reduced form?
Now they're reduced, absolutely. When we write the H's on it, now it's reduced. With the pluses, it's oxidized, not carrying electrons. When it's got the H, it's reduced. It is carrying electrons that it picked up from the food. NAD and FAD are used in respiration and metabolism that we're going to talk about. The P's, NADP and NADPH, are in photosynthesis. P for photosynthesis, remember it that way. So you won't see NADP and NADPH anymore after this slide because they're part of photosynthesis. We're going to focus on NAD and FAD because they're used in respiration. We good with that? Next ingredient that you need is enzymes. Enzymes are biological catalysts. They're molecules that make reactions happen faster. Let's get rid of our electron carriers here. And let's focus on respiration. What happens when we eat the sugar, when bacteria eat the sugar and break it down? This reaction would take days, weeks, possibly months without enzymes. Let's say you had a donut for breakfast this morning full of sugar, right? It would be in your stomach for a very long time if you had no enzymes in your stomach. Okay? But we've got tons of enzymes to make these reactions happen faster, so it'll be digested in a couple of hours instead of a couple of days or a couple of weeks. So it makes a real difference on time. They really speed up reactions. Now, they're very, very specific. There's not one magic enzyme that makes all chemical reactions happen faster. That would be nice, but it's the other way. Every chemical reaction has its own enzyme that makes it happen faster and nothing else. So this is our basic reaction again. A plus B yields C plus D. We got a name change here. Substrates. Substrates are the chemicals present before the reaction occurs. Remember a few slides ago, we just called them reactants, right? Chemicals present before the reaction occurs. Now they're substrates, because if you're using that word substrate, you're telling whoever you're talking to, right, that there's an enzyme being used. And when we just called them reactants a couple of slides ago, it could have been happening in a beaker all on its own. There's no enzyme involved. So if you're calling them substrates, you're saying there's an enzyme being used to speed up this reaction. Definition, though, is the same. Substrates are the chemicals present before the reaction occurs when an enzyme is involved.
You said substrates is what?
Chemicals present before the reaction occurs when you're using an enzyme. And if there's no enzyme, you just call them reactants. All right, so let's stick with A plus B yields C plus D.
So all of these components have to be present, every single one of them?
Or metabolism is not gonna happen. Okay, so let's say that that's what chemical A and chemical B look like. And the products C and D are gonna look like that. So that's the reaction that we're gonna do. There's an enzyme that is specific for this reaction. Kind of like Pac-Man. Imagine it as a ball, right, 3 dimensions. And it's got a sort of pie-shaped piece cut out of it. That is the active site. And notice how nicely our substrates look like they fit in there. Right? That's on purpose. Enzymes are specific for one reaction. This enzyme only works for this reaction because the substrates fit. Other chemicals aren't going to fit in there, so other reactions won't be sped up by this process. So the active site, that's the physical location on the enzyme where the substrates bind. The substrates are actually going to fit in there. So to draw that picture, plug A and B in. They will fit in the active site. The reaction happens right when they're there. So next picture. The enzyme is now empty, and products C and D are floating around. So the enzymes bind to the substrates at the active site, the reaction happens, and the product is released. And the enzyme is not used up or changed in any way in that process. Right? The enzyme looks just the same at the end as it did at the beginning. So we can use that one enzyme over and over and over to speed up this reaction lots and lots of times. But it will only work for this reaction. It won't work for any others. So online class doesn't get to hear me or see me draw. Right, so here's that picture. The enzyme is the active site in perfect shape for those substrates. They bind, the reaction happens. This reaction separates those two subunits. The products get released, and the enzyme looks the same as it did over there.
So they start off separated, they join together.
It's just this particular example that they started off joined together, and at the end they were separated. But different reactions are going to do different things. Right? Plants were doing photosynthesis. They'd start with individual subunits, and then when we were done, they'd be joined together. Right? And then we eat the sugar and we digest it into individual pieces. So it just depends on what reaction